Hello guys, welcome to another video. In this video we are going to learn about a biotechnology course, that is gene regulation and expression. In this video we are going to learn about some operand systems related to gene expression. So what is gene regulation? Gene regulation includes a wide range of mechanisms that are used by cells to increase or decrease the production of specific gene products. We know that gene expression or gene regulation is two types. First one is prokaryotic gene regulation and eukaryotic gene regulation. So, prokaryotes do not need to transcribe all of their genes at the same time. They can conserve energy and resources by regulating their activities, producing only those genes necessary for the cell to function. In prokaryotes, DNA binding proteins regulate genes by controlling transcription. An operon is a group of genes that are regulated together. An example is the Lac Operon. This operon, that means this group of three genes, must be turned on together before the bacterium can use lactose as food. When lactose is not present, the DNA binding protein called Lac repressor binds to a region called operator, which switches the Lac Operon off. Then when lactose binds to the repressor, it causes the repressor to fall off the operator, turning the operon on. So this is a prokaryotic gene regulation. Then in case of eukaryotic gene regulation, transcription factors are DNA binding proteins. They control the expression of genes in eukaryotes by binding DNA sequence in the regulatory regions. Complex gene regulation in eukaryotes makes cell specialization possible. The process by which microRNA molecules stop mRNA molecules from passing on their protein making instructions is RNA interference. RNA interference technology holds the promise of allowing scientists to turn off the expression of genes from viruses and cancer cells, and it may provide new ways to treat and perhaps even cure diseases. So this is eukaryotic gene regulation. So now we are going to move to the operating system or operator. Firstly, we need to know some genetic control development before to go to the operating system. Genetic control of development such as homotic genes. So what are homotic genes? Homotic genes are master control genes that regulate organs that develop in specific parts of the body. Homobox genes share a similar 130 base DNA sequence called homobox. They code for transcription factors that activate other genes that are important in cell development and differentiation in certain regions of the body. Then Hox genes. Hox genes are group of homobox genes that tell the cells of the body how to differentiate as the body grows. So these are the genetic control of development. Now we are going to learn about the advantages of gene regulation, such as a number of related genes required for a particular metabolic activity can be switched on or off simultaneously. Then gene regulation enables the cell to adjust metabolism as per requirement of environmental changes and development. It is economical as it synthesis enzymes only when required. Gene regulation helps in growth and differentiation. It is helpful in smooth completion of chain reactions. So these are the advantages of gene regulation. Let's move to the operating system or operand system. Operand systems are common in prokaryotes. The first operon, Lac Operon, was discovered by Jacob and Monod in 1961. Later on, a number of such operons are discovered in such as Trip Operon, Rna Operon, His Operon, Volume Operon, etc. We know that operon is two types, inducible and repressible. So let's go to the inducible operand system. Here we can see the picture of inducible operand system, and this is a Lac gene, then cap binding site, promoter, then structural gene, and these are the messenger RNA. In first case we can see that Lac genes strongly expressed when low glucose and lactose available. Then in the second case, Repressor protein bind, then Lac genes are not expressed when Repressor proteins are bind. Here high glucose and lactose unavailable. Then in third case, when CAM gene activate, Lac genes not expressed. Then in case of low glucose and lactose unavailable. Then cap binding site in last case, very low basal level of gene expression, high glucose, lactose available. So we are going to learn this in briefly in next session. So let's start it. First on inducible operand system or Lac Operon. Here we can see Lac Operon contains regulator gene, promoter gene, operator gene, structural gene. So let's get started individually. First one is structural gene. Here you can see these are the structural gene, ZOA, structural gene. So they are those genes which actually synthesis mRNA, and mRNA controls metabolic activity of cytoplasm through the formation of protein or enzyme over the ribosomes. An operon has one or more structural genes. The lactose or Lac Operon of Escherichia coli contains 3 structural genes, ZOA. So these are the structural genes, ZOA. Then operator gene. Here you can see O gene. O gene means operator gene. It is a gene which directly controls the synthesis of mRNA over the structural genes. That means operator gene controls synthesis of mRNA over the structural gene. Then it is switched off by the presence of a repressor, and inducer can take away the repressor and switch on the gene. The gene then directs the structural genes to transcribe. Operator gene of a Lac Operon is made only 27 base pairs. Then promoter gene. Here you can see the promoter gene. Here P indicates the promoter gene. It acts as an initiation signal which functions as recognition center for RNA polymerase, provided the operator gene is switched on. RNA polymerase is bound to the promoter gene when the operator gene is functional. The polymerase moves over it and reaches structural genes, perform transcription. Here we can see the promoter gene and RNA polymerase. Then let's next part is regulator gene or lac I gene. Here we can see the regulator gene, or which indicates I indicates the regulator gene. In Lac Operon, it is called I gene because it produces an inhibitor or repressor. That means regulator gene produces inhibitor or repressor. The repressor binds to operator gene and stops the working of the latter. It exerts a negative control over the working of structural genes. Then repressor. It is regular protein synthesis all the time by the regulator gene I gene. Repressor is meant for blocking the operator gene so that the structural genes are unable to form mRNA. It has two allosteric sites, one for attaching to operator gene and second for binding to inducer. After coming in contact with inducer, the repressor undergoes conformational change in such a way that it is unable to combine with the operator. The repressor of lactose or Lac Operon is a protein having a molecular weight of 1,60,000. It is made up of 4 subunits, each having molecular weight of 40,000. So this is about the repressor. Then inducer. It is a chemical substrate, hormone, or some other metabolite which after coming in contact with the repressor changes the latter into non-DNA binding state so as to free the operator gene. The inducer for Lac Operon of Escherichia coli is lactose, actually allolactose or metabolite of lactose. So this is inducer. Then cap gene or cap protein. It is activator called catabolic activator protein. It exerts positive control in Lac Operon because in its absence RNA polymerase is unable to recognize promoter gene. Here you can see the cap gene. It is its gene located away from the operon, but the receptor cap sites occur near the Lac promoter. Cap activates Lac genes only when glucose is absent. RNA polymerase is recognized by promoter gene. It passes over the free operator gene and then catalyzes transcription of mRNAs over the structural genes. The mRNA pass into the cytoplasm and form particular proteins or enzymes. Out of the 3 enzymes produced by Lac Operon, lactose permease is meant for bringing lactose inside the cell. B-galactosidase lactose breaks lactose into 2 components, glucose and galactose. The enzyme like lactose or 3-galactosidase, which is formed in response to the presence of its substrates, is often called inducible or adaptive enzyme. So this is about the cap gene, and this is all about the inducible gene expression. Now we are going to learn about repressible operand system. Repressible operand system is commonly found in anabolic pathways. The operand is active and the enzymes produced by its structural genes are normally present in the cell. The functioning of the operand is stopped when the concentration of an end product crosses a threshold value. Here we can see repressible operand system. Here is the structural genes, operator, promoter, regulator genes. So let's get started. First one is structural genes. The genes are connected to transcription of mRNAs. Here structural genes E, D, C, B, A, structural genes. The mRNAs translate their coded information in the synthesis of polypeptides. polypeptides give rise to proteinaceous substances including enzymes. Tryptophan operon has 5 structural genes such as tryptophan EDCBA. Here you can see EDCBA structural genes. They form enzymes for 5 steps tryptophan synthesis. So this about the structural genes. Then operator gene. Operator gene, here you can see operator. O includes the operator gene, indicates the operator gene, and it controls the functioning of structural genes. Normally it is kept switched on because the repressor produced by regulator gene is unable to completely block operator gene. The operator gene is switched off when a co-repressor is available along with repressor. Here you can see the operator gene. Then promoter gene. It is the site for initial binding of RNA polymerase. The latter travels from promoter gene to structural genes provided operator gene is switched on. Here you can see the promoter gene. P indicates the promoter. Then other regulatory region components. 2 components of regulatory region occur between operator gene and structural gene. They are ladder sequence and attenuator. Here you can see the L indicates ladder sequence and A indicates attenuator. The ladder sequence is involved in controlling attenuator. Attenuator helps in reducing tryptophan synthesis when it is present in sufficient amount without switching off the operon. Then regulator gene tryptophan R. It forms, here you can see regulator gene R indicates the regulator gene. It forms a proteinaceous component for possible blocking the activity of operator gene. Regulator gene of tryptophan operon lies at a distance away from the remaining operon. Then aporepressor. Here you can see the aporepressor. It is a proteinaceous substance synthesized by regulator gene. Aporepressor forms a component of repressor for blocking the working of operator gene. For this, it requires a co-repressor. Here you can see co-repressor. When the latter is not available in proper strength, the operator gene is kept switched on because by itself aporepressor is unable to block the working of operator gene. Then co-repressor. Here you can see the co-repressor. It is a non-proteinaceous component of repressor which is also an end product of reactions catalyzed by enzymes produced through the activity of structural genes. The end product is often utilized in some other reactions so that it rarely accumulates and hence does not function as co-repressor. However, whenever it accumulates or becomes available from outside source, the end product becomes co-repressor combined with aporepressor. Forms repressor and blocks the operator gene. Structural genes now stop transcription. The phenomenon is known as feedback repression. It exerts negative control in tryptophan operon. Tryptophan amino acid functions as co-repressor. So this is all about the repressor operon system. Now we are going to learn about the differences between induction and repression. First difference is induction. It is the switching on of an operon which normally remains turned off, and repression is it is turning off an operand which normally remains switched on. Then second difference is induction is caused by a new substrate which is to be handled and metabolized, and repression is caused by increased formation or availability of a metabolite. Then third difference is it is generally connected with a catabolic pathway, and repression is anabolic pathway. Then fourth difference is the regulatory gene of an operand liable to undergo induction produces a repressor that blocks the operator gene, and the regulatory gene of an operand liable undergo repression produces a part of the repressor called aporepressor. The same cannot block the operator gene. Then fifth difference is induction is the removal of the repressor of an operand by the inducer metabolite, and repression is the blocking of the operator gene of operand through a complex repressor that is formed by union aporepressor formed by regulatory gene, and co-repressor which is actually a product of anabolic pathway. Then sixth difference is inducer is substrate, hormone or its byproduct, and repressor is a compound formed by an aporepressor and a co-repressor which is commonly an end product of metabolic pathway. Then number 7 is it brings about transcription and translation, and repression stops transcription and translation. So this is all about the gene regulation operand system, induction operand system and repression operand system, and what are the difference between induction operand system and repression operand system. So that's all for today. See you in the next video. Thanks for watching.